First of all, before I start talking about microRNA, microglial cells, and neuroinflammation, which is, I believe, there is always component of any type of pathological events in the CNS, I want to say thank you for invitation. And actually, I took a very good, very nice scenic trip from Frankfurt Airport to here along the Rhine River. And I saw a lot of history, a lot of castles, a lot of small villages. Unfortunately, when you drive a car in the US, you don't see that much history. But I can tell a little bit history about Harvard University. And this is actually Harvard Yard. It's the center of the Harvard University. And this is a statue of John Harvard, the founder of the Harvard University. But what actually it's known, when you walk there, you always see the parents that they make a picture touching the foot of John Harvard. You can actually see in higher magnification, the foot is shining because so many people are touching there. But actually what most of the parents don't know, but I know because I've been there for like almost 4 years, after half a year when their kids get accepted to the Harvard, during initiation party, they need to run naked in the Harvard Yard, and some of them urinate on the same food. So it's about some tradition, but it's... So of course this is not only tradition at Harvard, of course there's a lot of traditions about research, and our department is traditionally interested in neuroinflammation, and particularly Center for Neurological Disease is interested in 2 diseases, multiple sclerosis and Alzheimer's. So most of my work is dedicated to MS, but probably you will see from my talk that a lot of applications and a lot of questions have been asked are general for any type of neuroinflammation and neurodegenerative diseases. So I'll tell you briefly what is MS. So it's a chronic inflammatory disease of the CNS and affects young adults. There are a lot of neurologic symptoms, including paralysis and paresis. And pathologically, you can find in the CNS a lot of inflammatory lesions, more than one. And usually these lesions can be found in the white matter, and you can see the areas of demyelination and neuronal damage. So some of the cells called oligodendrocytes that rub axons, neuronal axons get damaged, and then you have basically naked axons or even axonal transections, and you have a lot of symptoms. And still, almost nothing is known about pathology, etiology of this disease. What is the cause? So presumably it's autoimmune or viral. Here's actually a map of MS. It always amazes me that this is disease of the northern hemisphere. It's not. It's basically to get prone to have high incidence of disease, you have to live up north from 37 degrees of latitude. Actually, it's true for the south, too. You have to live to the south from 37 degrees of latitude. You don't have this disease in close to equator. Why? Nobody knows still. But actually, this picture always makes me believe that there is some environmental factor for this disease. And if we know a little bit better about pathology of this disease, we might be able to basically prevent it, not only treat, but also prevent it. Although our department deals a lot of MS patients, to study basic mechanism, you need animal models. So we use experimental autoimmune encephalitis, or EAE. The good thing about this disease, it can be induced in a variety of animal strains. It can be induced in rodents and primates. It's really relevant to MS. You have also autoimmune T cells, which recognize some of the proteins of oligodendrocytes. It could be actually 3 main antigens are myelin oligodendrocyte glycoprotein, proteolipid protein, and myelin basic protein. Actually, we're going to use this model, which is common for B6 mice. And this model is also very good to use various knockout animals. Also, you have a progressive paralysis, which we can basically score. There is a very good scoring system and also very clear symptoms. And also you have focal areas of inflammation and demyelination. So I just give you some information. There are 2 ways how you can induce EAE. You can either take autoimmune T cells, for example, from MBP TCR transgenic mice. Most of the cells recognize MBP self-antigen in the CNS and then transfer them into recipient mice. So we can immunize mice with basically MOC and CFA subcutaneously, but you also need some to give them pertussis toxin. The reason why I need to give pertussis toxin, people believe it might open blood-brain barrier to initiate pathological events in the CNS. This typical disease course. Most of the time when they induce disease in mice, they recover. Sometimes they don't completely recover, but sometimes there is a complete recovery. But most of the time, disease is self-limited. So you always have, in the beginning, you have a preclinical period when mice show no symptoms. It's usually several days after immunization. Then you have a disease onset. Usually it starts with a limp tail. Tail doesn't move. If you take a regular mice, you can move tail very well. But actually here, you can see the tail is not moving, but actually then symptoms are progressing, and you have actually paresis and paralysis. You can see actually mice are crawling. And then after that, mice spontaneously recover. Sometimes they still have some remaining symptoms. Sometimes there is a complete recovery. And actually, we've been interested in microglial cells. And actually, what are the microglial cells? There are several views in this actually question. One view is a view of immunologist, is a CNS fixed macrophage system. So we have macrophages everywhere. We have them in the blood. We have them in many tissues. For example, we have them in the liver, called Kupffer cells. We have them in the skin. It's called Langengarde cells. We have them in the blood, called monocytes. And there are, of course, macrophages in the CNS, called microglial cells. And they do express a lot of specific markers, like CD11B. They also express a low level of activation markers, like MHC class 2 and costimulatory molecules. They also express a low level of CD45. It's also a very good marker of CNS-resident microglial cells. And they can rapidly respond to neuronal tissue damage. Of course, there is another point of view of microglial cells. It was defined by basically Ortega. He actually thought that there are 5 types of the CNS: neurons, astrocytes, oligodendrocytes, microglial, and synaptocytes, actually. And synaptocytes, probably you, in this department, you're more interested. And here we have some interest in microglial cells. And of course, why microglial cells are important? In fact, when we started our research about 10 years ago, many people thought that they're not really important. They just passively located in the CNS. Of course, there is some kind of activation, but many people thought it's bystander effect. But recently, actually, over the last 10 years, a lot of actually papers and data starting to accumulate that really microglial cells, they have a strategic location in the CNS. They activate within hours after neuronal tissue injury. Activation is associated with many diseases, including neurodegenerative disease like Alzheimer, exposure to toxic agent, traumatic injury, infection, or autoimmunity. Actually, they may be involved in epilepsy. What we've been really interested in other recent studies is how microglial cells interact with other cells within CNS microenvironment. As you can see, microglial cells, they interact with neurons, they interact with glial cells, and there are a lot of events going on there. And we try to at least to uncover some of these events to see what's going on in normal CNS and during EAE. There are some difficulties, and microglial cells are not always easy to distinguish from peripheral macrophages. So actually, if you take sections of the mice with EAE and stain them for microglial marker, or it could be EBA1 too, it does matter. So what you can see, there is a normal and affected part of parenchyma of the CNS. These are sections of the spinal cord. And you can see these microglial cells, they have all this morphology ramified with the processes. And usually they have very distinct phenotype. They have low level of CD45. But if you look at the lesion area, you have a lot of activated microglial cells and peripheral macrophages. There are a lot of macrophages in the periphery. And these cells can be different. They're not exactly microglial cells. And it's very difficult to take them apart. And one of the approaches that we used and has been used by other scientists before was the generation of irradiation chimera mice to distinguish all blood-derived cells from CNS resident cells. Here actually how it works. You take a donor mice. Donor mice can be labeled with some kind of reporter system. For example, they can be labeled with GFP. Although we use now different reporter system. We use MHC class 1 mismatched mice. We use special fractal kind, chema kind, which is expressed on only myeloid cells with link with the GFP. Or you can use just GFP transgenic mice. So you lethally irradiate your recipient mice, basically by giving high dose of irradiation. You basically wipe out all stem cells in bone marrow and take a bone marrow from the donor. And after 8 weeks of reconstitution, you basically have the mice which all resident microglial cells are GFP negative. And actually microglial cells are radiation resistant. That's why they, even after lethal radiation, they still stay in the brain. And all peripheral cells will be GFP positive. So basically in this system, you can easily take apart microglial cells from blood-derived macrophages, and then you can induce EAE in this disease and see what happens. Actually, we initiated this study almost 10 years ago. In the beginning, we wanted to see what happens with microglial cells versus peripheral macrophages in inflammatory lesions. So these are mice with EAE, macrophage. So blood-derived cells, peripheral macrophages are green. All macrophages are red, and activation marker is blue. Here's only 2 markers. You can see that all red cells are activated microglial cells. All yellow cells are peripheral macrophages. You can also see here at higher magnification. And if you add some activation marker, you can see all peripheral macrophages are activated, and also there are some activated microglial cells in the lesion area. I mean, these results actually clearly showed us that microglial cells are co-localized in inflammatory lesions with peripheral macrophages, and they have activated phenotype. Using of the sections has their own advantages, although sometimes it's very hard to quantitate the cells. And the best way how to do that is to use flow cytometry. But for flow cytometry, you need to isolate cells of CNS. Basically, here's a procedure that we do over and over again for many years. So first you do whole body perfusion. So the idea of this procedure, basically you replace all the blood of the mice with the PBS. And the reason why you need to do that, because the brain is highly vascularized and you have a lot of blood in the blood vessels. If you don't do that and you try to isolate cells of CNS, you're going to get a lot of cells from the blood, and you're going to get a lot of contaminating blood cells. Then you dissect the brain and spinal cord, then you homogenize it, and then you use a special gradient which you can isolate cells according to cell density. And most of all immune cells, including microglial cells, will be here in the interface. Then you can do multicolor flow cytometry. Typically, we use several colors. So we basically look at the several markers, up to 5 or 6, typically 3, 4. Okay, so the main question of this talk was how macrophages adapt in this microenvironment. We know that they're in the CNS. They know they participate in an inflammatory response. They're in the lesions. They're activated. But what makes them different? So there is a macrophage in the blood vessels, and there is also a macrophage in the CNS called microglial cells. What differ these two cell types? Macrophages can migrate to CNS in many events. There are normal events like development, when macrophages populate the brain early after birth. In mice, actually, there are 2 waves, before the birth and shortly after birth. And there are also pathological events. Any neuroinflammation is accompanied by migration of macrophages to the CNS. And Are these macrophages different from these peripheral cells? And this is fundamental questions many people try to answer. People try to find special antibodies which recognize specific markers, and actually they couldn't distinguish them. People try to do some proteomics. Also, it's very hard to distinguish. mRNA level, there are some results, but again, it's very hard to get them to the protein level or surface marker level. So we thought that adaptation to CNS microenvironment is a little bit more fine-tuning mechanism. It's regulated on a little bit different level. We thought it's regulated on a mRNA regulation level. So there are some specific proteins. Actually, I've been talking today about actually how it's called, this factor CP. Yeah, here we go. That can regulate mRNA of the target gene expression. And also, it's known that microRNA can regulate expression of the target genes. MicroRNA can be viewed as an analog of siRNA. So I can try to tell you a little bit more about microRNA. So if you never heard of microRNA, I can say microRNA is a natural analog of siRNA. So siRNA is artificial short RNA, which can be, kind of delivered to the cell, and it can target the mRNA and basically prevent it from, basically lead to destruction of the mRNA of the target gene. MicroRNA is a natural analog of siRNA. It's a grown family, and microRNAs are pretty small, about 20 nucleotides. There are about 1,000 of them. And it has been shown that they're implicated in many processes, especially cell death and proliferation and differentiation. A lot of similar between microRNA and siRNA, but there is a difference. What actually siRNA does mostly, siRNA mostly lead to degradation of mRNA of the target gene. So when siRNA binds to mRNA of the target gene, this complex gets degraded. And microRNA sometimes work this way. But most of the cases, microRNA doesn't lead to degradation of mRNA of the target gene. It rather leads to suppression of translation of mRNA of the target gene. So that's why probably it's important to know, if you add microRNA to the cell, you might see the difference in protein level expression, but you might not see much difference in mRNA level expression. I just want to summarize this, that siRNA is more important for basically turnover or degradation, whereas basically microRNA is important to repress translation of the target gene. Since microRNAs are natural analogs of siRNA, it has to be synthesized and has to be somehow be produced in a cell. And there is a very complex process of doing that. There are special genes which encode microRNA. It usually translated as a long precursor. Then there are specific enzymes which degrades this precursor. And then you have actually a very small duplex. In this duplex, you have one chain which is functional, can block translation of the target gene, and another chain is non-functional. So this is duplex. And this duplex actually is very stable and typically used to transfect cells. You can make synthetic duplex and transfect cells and do some functional studies. So we've been interested in microRNA-124. And this microRNA is also pretty short. It's about 21 nucleotide long. It's 100% homologous from mouse to human. And it's mostly expressed in the CNS, not in other organs. What is known about this microRNA? It's promote differentiation of neurons. It's really important for that. It's also important to repress repressor which block alternative splicing of non-neuronal genes. On the other words, what MicroRNA-124 does, it promotes expression of specific neuronal cells. It's basically inhibitor of neuronal genes. So basically, if you have miR-124, you get a lot of neuronal specific genes expressed in a cell. And actually it's important because, especially in the case of microglial cells, because microglial cells, they might express some genes which are similar to neuronal genes, like channel proteins. Also, miR-124 was important for tumor suppression to restrict proliferation of cells. Okay, so what we first did, in the beginning, we performed selective profiling of different types of microRNAs which are expressed in macrophages and in the CNS. We actually found that there are about 45 more different microRNAs. And we did a real-time PCR, and we found that one microRNA, miR-124, was really expressed in microglial cells, but not in other macrophages, which we isolated from blood. We isolated them from bone marrow, peritoneal cavity, liver. These are thioglycolate. It's inflammatory macrophages in peritoneal cavity. And these are bone marrow-derived macrophages, which can grow from stem cells with a specific macrophage colony-stimulating factor. So none of these cells express miR-124, but macrophages in brain and spinal cord express rather high level of this microRNA. This is the opposite microRNA, miR-223. This microRNA is mostly expressed in myeloid cells, and it was expressed in all peripheral cells, but not in microglial cells. And actually, that's also a very interesting result, because usually microRNA-223 is important for differentiation of myeloid cells. And from these results, we can basically see that microglial cells in the CNS, they are more mature as neuronal cells, but less mature as myeloid cells. And probably I can come back to this speculation later, but it's kind of point I wanted to stress. So, but as I said, it's very easy to distinguish phenotype and markers for microglial cells in normal CNS versus peripheral cells. But what's going on during inflammation? And during inflammation, the situation is much more complicated. As I showed you before, in the lesions, you have both macrophages and microglia. They're located in the same area. They might have similar properties. So what we did, we used these chimeric animals. So we isolated mononuclear cells from the CNS of mice with EAE. And as you can see, clearly in these chimeric animals distinguish peripheral macrophages. Down here, this is macrophage marker. So these are peripheral macrophages, and these are microglial cells. So we sorted the cells by cell sorter and look at the miR-124 expression by PCR and found that really miR-124 was expressed at much higher level in microglial cells when compared to peripheral macrophages. Okay, in next question, we wanted to see what's going on with miR-124 in microglial cells when microglia become activated. So we found that in normal CNS, microglial cells express miR-124 at a rather high level, but over the time course of EAE, this is onset, peak, and recovery, you can see that microglial cells downregulate miR-124. And what is actually another interesting finding we got here, that some of the peripheral macrophages that came from the CNS, they start express low level of miR-124 during recovery phase. And when we look at the activation markers of microglial cells versus macrophages, we separated them by the level of expression GFP. These are GFP negative, these are GFP positive. So you can see in NOD disease, microglial cells are not activated. They express low level of CD45, they're negative for MHC class 2. Then during onset, microglial cells become activated, even more activated during the peak of disease, and during recovery, microglial cells came back to non-activated states, at least partially. And what about peripheral macrophages? Peripheral macrophages came to the CNS as very highly activated. They still activated during the onset of disease. But what is interesting, during the recovery stage, you have some subset of the cells that downregulate all activation markers. We kind of think that these cells, they start adapting in this microenvironment, especially during the long recovery process, and become microglial-like, and even start express some MIR124. We found that MIR124 expressed in microglial cells, but what does it do to these cells and to macrophages? So we took bone marrow-derived macrophages, which don't express MIR124, and these cells in culture, they have highly activated phenotype. They express high level of various activation markers. But when we transfected the cells at MIR124, they downregulate all activation markers, including MHC class 2, F8, and CD11B, as you can see here. We also found that transfection MIR124 downregulate expression of TNF-alpha, which is a very potent pro-inflammatory cytokine, and which is very harmful for neurons. And in this experiment was also very interesting. We found that transfection of MIR124 skew macrophages from pro-inflammatory to anti-inflammatory phenotype. It is known that there are two types of macrophages. One's called M1, or pro-inflammatory. They express a lot of TNF-alpha and NO, which is harmful to neurons. And M2 macrophages, they express anti-inflammatory cytokines like TGF-beta or arginase. And you can see when you transfect macrophages with MIR124, these are all actually green. All this green, it's MIR124 transfected. So you can see there is downregulation of INOS. These cells produce less NO, but they start produce a lot of anti-inflammatory cytokines and arginase. Arginase is an enzyme which is opposite to INOS, and it's also important to metabolize arginine. But here, usually, these when INOS metabolize arginine, you get you get NO production. Here, there is no NO production. So, which means MIR124 makes macrophages less harmful to the CNS and less inflammatory, more anti-inflammatory, and more get to the phenotype which promote tissue repair, we believe. Okay, so to conclude this part of the talk, I can say that MIR124 expressed in microglia, but not peripheral macrophages. Activated microglia downregulate MIR124, and the overexpression of MIR124 result in deactivation of macrophages in vitro. Okay, here it's about mechanism, how MIR124 result in deactivation of macrophages. We perform in silico analysis of different downstream targets for MIR124. Actually found one very good candidate. It's called CBPA. We found that there are 3 sites for binding of MIR124 in this protein. And CBPA is really important because it's a master transcription factor which is required for differentiation of macrophages. What we found that there are 3 binding sites, as I said. These sites are very conservative between various species. This is a 3' UTR of a CBPA gene. And when we did some reporter, luciferase reporter construct, here so we have a luciferase gene and a 3' end, we put a 3' UTR of a CBPA gene. And we did several mutations of MIR124 binding sites. So here, this is just control reporter system when you basically, there is no luciferase here, just a vector. But when you transfect cells with this reporter plasmid, you have some level of luciferase. But when you add MIR124, the level of luciferase goes down, which means translation of this CBPA, which means actually there is a blockage of translation of this construct, went down. But when we mutated 2 sites, which actually close to each other, you can restore basically the level of expression of this construct of luciferase. But if you mutate all 3 sites, you basically get expression of luciferase to the normal. So basically, you remove all this inhibitory effect of MIR124 expression of this gene. It was very good actual data. It's really proved in vitro that MIR124 targets CBPA alpha. Okay, so as I said, it's a master regulator of myeloid cells. And CBPA alpha induced another factor which is called PU1. And both of these factors really are required to differentiate any cell type to macrophages. Without these factors, cells wouldn't become macrophages. CBP alpha also acts in cooperation with NF-kappa B in inflammatory response. So basically, there are a lot of studies, including CBP alpha and PU1. And if you have knockouts of these genes, you have markedly diminished monocytes and neutrophils. And basically, if you transform fibroblasts with CBP alpha and PU1, you can transform the cells into macrophages. And microglial doesn't express CBP alpha in normal CNS, but it can be induced after activation of these cells. It was actually very important. So here we show that really CBP alpha was downregulated. Usually it's like from 2 to 3-fold difference by miR-124. And we also confirmed by intracellular flow cytometry that CBP alpha goes down in macrophages transfected with miR-124. The same thing for PU1. So basically, miR-124 downregulate both these factors. And the next experiment, we wanted to move our study to in vivo effect of miR-124 on EAE. So what we did, we injected liposomes with a miR-124 IV every other days in mice with EAE. And what we found was very interesting, that in the spleen, actually this is real-time PCR, miR-124 really was significantly upregulated when you inject the liposome with miR-124, but it's not in the dendritic cells, not in the B cells and CD4 T cells, not in other cells in the spleen. Mostly macrophages picked up miR-124. Actually, to some extent of degree, it's not surprising because it's known if you inject liposomes, usually it's engulfed by macrophages because these are phagocytic cells. We did similar analysis with the fluorescently labeled miR-124, and actually what we found that you basically get some of fluorescently labeled miR-124 in macrophages, dendritic cells, and also have to some extent degree in microglial macrophages in mice with EAE. In mice with EAE, blood-brain barrier is open to some extent of degree, so we are not surprised that miR-124 went there. It was a little bit strange with dendritic cells because when you look at fluorescently labeled miR-124, it's in dendritic cells, but when you try to compare it by PCR, it wasn't there. So we thought that there is some specific degradation of this microRNA in dendritic cells. So it's kind of good example to show that not always you have a functional transfection of the cells, especially in vivo. Here you can see definitely macrophages are transfected, but other cell types not really. This is the effect on the clinical score. This is a control. You have nice EAE curve. And when you start inject miR-124 starting from disease onset, we did it every other day, IV. So you can see that disease basically went down pretty dramatically. These are some pathological events in the CNS in this treated versus non-treated mice. And we really found that we have lesions in the CNS of a control group, but nothing in miR-124 treated mice. And what we also found that there was a big decrease in the number of infiltrating lymphocytes, T cells, and activated macrophages, microglial cells in the CNS. We also found that after this treatment, you have deactivation of macrophages in the periphery, in the spleen. So we thought that this clinical effect of miR-124 when it was injected IV was due to systemic deactivation of macrophages in the periphery and to some extent of degree in the CNS. It's a little bit busy slide, but I don't want to give a lot of details. It just shows that really after treatment of miR-124, you got a decrease in level of cytokines on T cells. These are autoimmune T cells. You got less interferon gamma. They proliferate less. This is BRDU, which measure proliferation of the cells. Less of activation markers. The same true for macrophages. They produce less TNF-alpha and IL-6. These are both pro-inflammatory cytokines, which really upregulated significantly during any type of inflammation. So basically, all these cytokines went down when you treat mice with the miR-124. So to To summarize this part of my talk, I'll tell that miR-124 repressed CBP alpha. It's a downstream target for this microRNA. Systemic injection of miR-124 resulted in substantial amelioration of EAE symptoms and systemic deactivation of macrophages. Probably it's the last part of my talk was concerning the interesting question that we started with, how expression of miR-124 is regulated in CNS macrophages, so particularly in microglial cells. So hypothesis was that macrophages may receive some signals from other cells in the CNS. We thought mainly from astrocytes and neurons. And the way we addressed it was to co-culture macrophages with the neuronal and astroglial cell lines. We also did some conformation in vivo. So if you take regular bone marrow macrophages, they have rather activated phenotype, but when it's measured by MHC class 2 expression CD45, it's here. When you co-culture with astroglial line, you can see they downregulate activation markers. And you get even stronger effect when you co-culture macrophages with a neuronal line. And actually, when you try to analyze what happened to these cells, we sorted them from co-culture, of course, to make sure that there is no contamination here of astroglial cells and neuronal cells and macrophages. When we sorted macrophages in this single culture or co-culture, what we found that if you take just bone marrow macrophages, they don't express miR-124. But if you co-culture them with astroglial line, there is some upregulation of miR-124, or even much better effect than a co-culture neuronal line with the macrophages. Interesting that neuronal line express miR-124. It's rather high level. Here is a comparison to adult microglia, which have very high level of expression. But astroglial line, they don't express miR-124, although they can induce it in macrophages. So there is crosstalk between actually neuronal glial cells with the microglial cells, actually, we believe in normal CNS. And as I showed you, during EAE, miR-124 goes down. We think this crosstalk is disrupted during inflammatory response. These are also co-culture experiments when I try to use miR-124 inhibitors. In addition to overexpression of miR-124, you can inhibit it. There are special synthetic nucleotides. It's called LNA. These are synthetic analog of a nucleotide that binds particular microRNA and inhibit it. So basically, when we use inhibitors are not perfect. They never block it to 100%, although they might be effective, especially for in vitro studies. So we basically what we did co-culture experiment, and we got some downregulation of these activation markers. But when we add inhibitor, we kind of stopped this process. Here you can see just 15% of the cells have high level of activation markers, but here you have 37%. So you can basically block it. There are some changes in morphology. Here it's a co-culture with astroglial line. You can see the cells have some processes. When you add inhibitors, they kind of lose it. Not the best picture in the world, but you can see some effect here. It's much better when you quantitate it. So, and the same true for neuronal line. If you co-culture them, you have some changes in morphology. You can even see there's some contacts between neuronal cells and macrophages. Macrophages are green here. They're from GFP mouse. You can see them. And if you add inhibitor, you get kind of more round cells. Again, it's not the best picture, but you can definitely see here that there is some kind of abrogation of these co-culture experiments, co-culture effect of downregulation of activation markers. So we also did some in vivo studies, although these studies are a little bit complicated and a little hard to interpret it. Although we use here chimera mice, all microglial cells here are GFP positive. Peripheral cells should be GFP negative, so you can only see microglial cells here. So we injected intracranially just control liposomes, so anti-miR-124 inhibitors. And usually when you inject it, about like 20% of the macrophages pick it up. Actually, it's a much worse level of transfection when you do it by IV injection during EAE. And as you can see, that microglial cells, they become more activated here when compared to control. And it's the same true for if you do it by flow cytometry. So just to summarize this part of the talk, that co-culture macrophages with astroglial or neuronal lines resulted in upregulation of MIR124, an induction of microglial-like phenotype, we call it, and inhibitors resulted in activation of microglial macrophages in vitro and to some extent of degree in vivo. Here is the model that we want to establish. So we call it dynamic equilibrium. That in normal CNS we got microglial cells. If macrophage go to the CNS in normal conditions, we get some signals from neurons and astrocytes and upregulate MIR124, become more microglial cells. But actually, when some inflammatory cells come, especially it's true for AE model, but it's for other neurodegenerative models, you can basically remove some of the signals. If there are some pro-inflammatory T cells coming, they basically change the balance towards macrophages. And I think it's always a balance between microglia and macrophages, and it depends on these signals. I'm just going to tell you briefly about future directions. Of course, there are a lot of questions still we need to answer in this study. We still look at the mechanism, what MIR124 does with macrophages in terms of transcription factors, in terms of many actually pro-inflammatory and anti-inflammatory cytokines. Also, we try to develop a system to look at what's going on in mice with EAE in vivo. When injected in vivo with liposomes, we see how these liposomes go to different cell types, how they basically how to deliver in better way MIR124 to macrophages. You know, there are several ways you can do that. Recently, it was a report when people took basically siRNA and put them in a glucan shell. They took shells from a yeast, put siRNA there, and then feed mice, and actually delivered very successfully siRNA to macrophages. So there is a way like that to do it actually with MIR124. So we're still thinking about it. And delivery is always an issue about with the microRNA siRNA studies. Although we were lucky in our results, although it might not be the case if we try to translate it to human research. And probably the most interesting question, basically, what's the role of MIR124 in promoting quiescence of microglial cells in CNS microenvironment? And here, actually, we think that there are several possibilities how it might happen. We think that what happens, there is a possibility that there are specific factors, soluble factors, which are produced by neurons, might upregulate MIR124 in microglial cells. We think there are several candidates of TGF-beta and fractal kind. Also, it's possible that it happens, and we believe this is the case, that there are special cell-cell contacts and there are special synaptic-like structures. I wouldn't call them typical synapse or typical gap formation as, for example, glial cells and for neurons, but people usually call it neuroimmunological synapse. And there are several molecules which can be involved in that, and especially it's known that NCAM, neuronal adhesion molecule, which is expressed in microglial cells and expressed in neurons, might actually contribute to upregulation MIR124. And also, there's a very interesting hypothesis that microRNA can be directly transferred from a neuron to microglia through specific exosomes. Exosomes are small secreted particles. And actually, are known that there are a lot of microRNAs in these exosomes. Actually, we found some of we detected MIR124 in exosomal fraction prepared from a brain and from cultured neuroblastoma. When we can actually isolate them on a sucrose gradient and see that, but again, it's an interesting hypothesis that needs to be addressed in the future. So basically, I want to acknowledge people that helped me. The work was done in the Howard Weiner Laboratory, Harvard Medical School, in collaboration with Anna Krychevsky, Tatiana Verimaker. Actually, she's my girlfriend. She did a lot of experimental molecular biology, microRNA. She actually works in a microRNA in neurodegenerative diseases. Actually, she's more interested in long-term potentiation and MicroRNA 124. But actually, she helped me a lot with, to establish all these microRNA techniques. And we did some imaging in flow-in imaging core facility. So, and thank you for your attention. And I would be hope.